Okay, now we're on to 8.5, which is translation, the last stage of this the central dogma thingy. Okay, translation. Translation is a process that converts an mRNA message into a polypeptide. Polypeptide is, the same thing as protein, but I wrote protein in my worksheet and apparently I got that wrong. So just remember it as polypeptide. So translation is a process that converts an mRNA. So it's just the creation of protein. Now, what is a polypeptide? A polypeptide is a chain of amino acids. So it's essentially, many amino acids that are combined, stuck together, and which is the same thing as protein. But yeah, anyways, amino acids. These amino acids are combined, and they are used to create the protein that we use in our whole body. And the thing about amino acids are there are 20 different types, not 21, not 19, but 20. And it's not that there are 20 in a certain protein, but there are 20 different types. Like how there are different colors, there's 20 different types of amino acids. So these 20 different types together make a protein. And so amino acids make polypeptides, which make protein. Now, translation here, it occurs in the cytoplasm. And what else is in the cytoplasm? Ribosome, like I said before. Please remember that. Now, a ribosome is like a machine, a factory, like I said. It reads the message from the mRNA, and it goes like, oh, okay, we'll combine these amino acids together and produce protein. So essentially, it's a factory that, it's a car factory. It combines different parts of the car and produces cars, right? So the same thing here. Ribosome is a factory. It combines amino acids together and it produces protein. The product from the ribosome is protein. Okay, now there's this thing called a codon. Very important. It's a three nucleotide sequence that codes for an amino acid. Very complicated saying, however, not that complicated. Okay, it's a nucleotide. What is a nucleotide? In RNA, it's AUGC, right? These are the four types of nucleotides. And it's a three nucleotide sequence. So as you see here, this is a table of the types of codons. Anyways, these three, this is what we call the three nucleotide sequence. Three, 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 three. There are three nucleotides in this in a group, so it's a three nucleotide sequence, and there are many different types, and these are codons. So these code for certain amino acids. You see here, uracil, uracil, and uracil code for this thing called PHE, which is a name that you apparently don't need to memorize according to Miss Kim. However, there are some things that you have to memorize, but I'll talk about that later anyways. So if you see here, CAU, this is a codon, but C here is a nucleotide. Three nucleotides equals one codon, CAU, which also equals this thing, which is an amino acid, HIS, not Handong International School, no. HIS, this is an amino acid. Now, three nucleotides make one codon. Like I said, three nucleotides make one codon, which makes one amino acid. And these things are found in RNA. So in total, there are 64 different types of codons. So there are 61 codons. From the 61, one is a start codon, and there are three stop codons. And they code for 20 types of amino acids. If you count the amount, then there will be 20. I have never stopped to count, but there will be 20. I trust Miss Sarah. Anyways, they are read as a series of non-overlapping nucleotides, which means that they are read without spaces, so there are no spaces in between them. In a reading frame, we did this worksheet in bio where there were nucleotides in a row and as in a row. That's a reading frame, like AUCG AUGCCCGU. That's a reading frame, and it's a way to divide the sequence of nucleotides in that model into it's a set. It's like a set, a sequence, yes. And the order in which the nucleotides are read are very important. So if you change the order, then the protein changes.